Hi everyone. So, following the questions today, we're going to do a bit of a reprise of PAMPs and DAMPs and linking it through to inflammation. So in a minute, I'll sort of flip the camera around and we'll just do some drawing along. This isn't rehearsed or anything, and there might be some details that I missed, so I'd recommend also speaking to Michael Dillon, Alex Hughes, just hear what they have to say. We do have another session coming up and a bit more immunology on the way. If you want to look at a textbook, I'd recommend Janeways. It's called Immunobiology, I think, which has some excellent diagrams. My approach to all of this is just the kind of basic principles. So it's about not so much about loads and loads of details, but it's about looking at things in terms of a kind of like integrated approach. So we look at the immune system not just through the eyes of cells, but also the interaction with microorganisms and also other aspects of normal physiology. So the clotting cascade and the immune system, for example, overlap in lots of different ways. And that's something that sometimes can be missed. Let's just go through it and we'll just flip the camera. So hopefully you can see, there we go, piece of paper. So what we're going to do, let's go PAMP and DAMP. We'll kind of, along the way, we'll sort of define the terms and I'll try to write down any acronyms that come up. As a reminder, these are both umbrella terms for molecules found in and on microorganisms and typically in your own cells. PAMPs are produced or present in the body when a microorganism is present, and a DAMP is present when there's damage to a particular tissue. These are both detected by an umbrella term of receptors called PRRs. This stands for pattern recognition receptors. Why these umbrella terms? The bottom line is that there are just too many really to talk about. So there are loads of different PAMPs, there are loads of different DAMPs, and there are several different receptors and receptor types that can be used to detect. So you can kind of think about the surface of cells just being decorated in all sorts of different receptors. And an important thing to say is that these come and go. So your body can regulate the expression of these things. And so in some ways, different tissue types, different cell types within that tissue will display different receptor types at the time. And these are all looking for different things. So again, it's just about appreciating that your body has different ways of recognizing different internal and external molecules. Here we have a generic pathogen. And let's say we have a pathogen. What kind of specific molecules are we going to find? Well, thinking about bacteria, we're going to have peptidoglycan. We're going to have in gram-negatives, lipopolysaccharide, gram-positives, lipoteichoic acid. There are other specific molecules, so inner and outer membrane molecules. The flagella is made of a protein called flagellin, for example, which is again detected by the immune system. These essentially are molecules specific to microorganisms. Sometimes you might actually read this as a MAMP, and there is this is microbial associated molecular patterns, because of course not all microbes are pathogens. And what about DAMPs? Well, DAMPs essentially are molecules found inside cells where the only way they come out is when there's tissue damage. And it's these things that your body is detecting. Okay, so, and again, we think this whole thing is about triggering inflammation and about trying to simultaneously control infection, but also about about tissue and wound repair. So these are detected by pattern recognition receptors, and these are going to be the things sticking off on the surface of cells, which ultimately trigger a cytokine or vasodilator or some kind of signal release. And that's important at telling nearby cells and the rest of the immune system what's going on. Why do we do this? Ultimately, this is all about inflammation. And inflammation is a key normal physiological, but also a pathological process when inflammation goes out of control. And again, that's a kind of a key thing to say. You know, inflammation is kept in checks and balances. When this regulation goes wrong, that can itself lead to disease. So if we think about things like sepsis, when you have uncontrolled inflammation or systemic inflammation, you know, you'll get kind of massive disease caused from that. Let's think a little bit about some of those hallmarks. These came up before. And some of them that were mentioned were exactly right. So, you know, you'll get heat, redness, swelling, pain. And of course, these reflect the underlying pathology and what's going on. So we've got blood flow goes up. Vessels become leaky. I'll say loss because what's happening is that fluid is moving from one compartment to another. So it's moving from the kind of intravascular compartment, so inside blood vessels, into tissues where you've got either your microorganism has made an appearance or there's tissue damage, sometimes a combination of the two things. What's then happening? Okay, so let's just go into this in a bit more detail. And this is what I drew before. Imagine a barrier. We'll just call this a barrier. We'll just say maybe this is like skin. Let's consider there's a combination of things that have happened. So let's just consider there's been some damage, tissue damage, so trauma. And then because the skin is colonized by different things, we'll then have escape of microorganisms in as well. So we've got microbes. These are going to be the PAMPs, trauma, DAMPs. What's going to happen? Both of these things can be detected by endothelial or epithelial cells. And in turn, they can go on to trigger vasodilation or signal for cells to be recruited into the area. We mentioned earlier the presence of macrophages. These are a good example of an antigen-presenting cell. A macrophage is a tissue resident, so they are drawn in from blood vessels where they're referred to as monocytes, and they then differentiate into macrophages. And so macrophages are monocytes just found in specific tissues. You'll find some in the skin, found in the GI tract, found in the lungs. And both of these things can detect. So they can be detected, and they can be detected. Remember, it's a non-specific way of detecting trauma and a foreign invader. And then what happens? Well, a few different things can then happen. So the macrophage can then begin to signal, and there are a few system-wide things. So it can begin to recruit white blood cells, depending on the type of immune system, there are different white blood cells that can be attracted to different invading organisms. We'll come on to that in a second. But specifically, we're thinking about inflammation. So let's imagine we've got a blood vessel now that's become nice and leaky as a result. We start to see fluid moving out. Let's just consider this for a second. What happens? So fluid moving out of a blood vessel into a nearby tissue will result in swelling, edema. Why is that needed? Well, it's going to contain cells. So, well, firstly, it allows white blood cells can move out of these capillaries. Without the inflammation, they're trapped. So the white blood cells can move into the area. Soluble fluid will have things like complement. It will have antibodies. It will have clotting. I mentioned before, platelets. All of this is required. Okay, so the complement in itself can also then keep the inflammation going. It can also opsonize. And it can also form the membrane attack complex as well. So that's good. There's another thing that happens as well, and that is the blood pressure goes down because there's now less fluid in here. So the vascular resistance has gone down and your blood pressure also goes down. So what we also then see is things like your heart rate goes up. You'll also see your respiratory rate go up to compensate. And this is all about the fact that you've got fluid exiting. There's therefore less throughput, which means that there's less oxygenated or there's less fluid moving around the system. And then, as I mentioned, up here, you can't see it, but up here, we then got a few white blood cells that are being attracted as well. So for example, if we had a bacterial infection, we might expect to see neutrophils being recruited. There are a few other markers as well. So again, up here, I mentioned that your macrophages are going to be signaling to the system. What does that mean? Well, that could mean, or that could indicate, it tells your liver to start to overproduce, for example, things like clotting factors. Your liver is also the site of complement proteins being made. It's also the site of a molecule called CRP being made as well, the C-reactive protein. So this is the thing. This is what we're dealing with here. So there's obviously a lot more that we could kind of go into. But in essence, you've got PAMPs and DAMPs are generic, or well, they're generic terms for a whole bunch of different molecules detected by the immune system to trigger a response, which elicits a whole different range of things happening. All of this is required to allow white blood cells and soluble factors in to begin this process of simultaneously controlling infection, but also tissue repair. I mean, an aspect that I didn't even talk about is the fact that macrophages aren't just there to gobble up foreign invaders, but they themselves can have a role in directly affecting tissue repair itself. Some of the monocytes coming in can or have on their surface tissue factor, which can help to promote clotting. So there's a whole bunch of overlaps here. As I said, all these things, please do quality check and go back and have a look at all this stuff. And if I actually just try to zoom out slightly, you might even, we'll finish here on a slightly, or on the final version. I'll see if I can take a picture and send it on to you. How was that? Hope that was all right.